Okay, class, we're going to call this lesson 23. This is from the lecture 18 PowerPoint slides. And lecture 18 is entitled Prokaryotic Gene Regulation 2. And then this covers the tryptophan, the arabinose, and the lambda phage um, systems in um, these are prokaryotes. And then lambda phage is a virus. So, prokaryotic gene regulation, part two. Okay, so we left off talking about the lac repressor, and I'm just showing you the slides. I don't want you to memorize this. I'm just showing you this is where the location of these genes are. So this is the end of the lac I gene. Remember, it's called lac I because it's inducible system, but it actually is a repressor, and that repressor binds to the operator, which is located right there. So here's the end. This is the stop codon, right? That's the end of it. And then this is the cap cyclic AMP binding site. This is part of the transcription factors that help recruit the polymerase that we already talked about. And then this is the promoter sequence. Here's the -10 and the -35 box that we've discussed. This is the operator. This is where the lac repressor, so this product binds here. And it effectively blocks the polymerase from doing, and remember that's an RNA polymerase, from converting this DNA into RNA. And then this is the start of the Z. Remember it's ZYA. This is beta galactosidase. This is permease. And this is, and who cares? So, anyway, that's what's going on here on a DNA level. So you can kind of see it. Again, I'm not asking you to memorize this. I just want you to see what this looks like on a DNA level, a sequence level. Okay, so the next thing we're going to talk about is the tryptophan operon. So in lactose, it's an inducible system, right? You don't want to turn on the genes to break down lactose unless lactose is present. So lactose is what we call an effector because it affects the gene. That's for the lac operon, not for this one. Um, so in this case, if lactose is absent, uh, not in this case, but the previous slides we were talking about, lactose is absent. We don't need the lac genes to break it down, so it's off. And remember, it's also regulated by glucose. If lactose is present, we need to turn on the genes to break down lactose so that we can use it as an energy source. So that's why it's an inducible system. The effector lactose induces the gene to turn on. All right. So that's why it's called lac because it's an inducible system, even though that's a repressor. Okay, so in tryptophan, this produces the amino acid tryptophan. Right? To produce tryptophan, you need 5 genes. So remember in lac, we had 2, Z and Y are required, A is not required because we can mutate that and it doesn't affect lactose metabolism. Here we have E, D, C, B, and A. I don't care that you know what those code for. I do care that you know what the lactose ones are code for. So remember, Z is for beta galactosidase that breaks that beta bond in lactose. And then this is permease. This is beta galactosidase gene that cuts that bond. Okay, so I just want you to know for this that there are 5 genes involved in it, that they're downstream, so they're structural genes. They convert produce the amino acid tryptophan. Okay, so tryptophan again is an operon because it's one single mRNA. Each of these have their own Shine-Dalgarno sequence and their own stop for translation. So they're producing 5 different products. I'm just going to make up shapes for these products. Okay, and these are enzymes, and they work together to produce tryptophan in a biosynthetic pathway. So TRIP-R is called R this time because this is a repressible system, not an inducible system. Again, TRIP-R has its own promoter, which is over here, it's not shown, and that affects regions in the regulatory area of the tryptophan. So this little break line is showing you that they're not next to each other, that this is further away, further upstream than the tryptophan operon itself. This is the promoter for tryptophan. This is the operator, so that DNA sequence that this is going to bind to. TRIP-R will bind to the O region. And then TRIP-L, it contains what we call attenuation. So there's a backup system for this, just like, there's a backup system for lactose, because if you have glucose present, you don't want to turn that on. You'd rather use glucose than lactose. So you don't want to make the genes to break down lactose. This one has kind of the same double way to ensure that this is not being expressed unless we need to make tryptophan. So this is the exact opposite of the lac operon, because here we want to turn on the genes to make tryptophan when tryptophan's absent. So in this case, we have tryptophan is absent, we want it on. If tryptophan's present, we don't need to make more tryptophan, so this operon is off. And this is exactly opposite of what lactose is. So lactose induces the gene's expression, it turns it on. In this case, tryptophan represses the gene's expression and turns it off. So that's why it's called a repressible system. Okay, so for the test, you should know there's a promoter, an operator, an attenuation region that's called TRIP-L, and that there's 5 genes involved in this biosynthetic pathway to generate tryptophan. That the repressor is upstream, and it codes for a repressor that binds to the operator, just like LAC, and it also binds to tryptophan. So we'll talk about exactly how this works in a second. Okay, so if tryptophan is present, you don't want to turn the gene on to produce tryptophan, right? You already have tryptophan, you don't need more tryptophan. So the presence and the binding, right? So remember lactose bound to lac-I. So tryptophan's going to bind to I guess you'd call it a repressor in tryptophan's control. Okay, so if tryptophan binds to the repressor, when we add one molecule to another molecule, it changes its shape. Just like if I took a Lego block and put it on my Lego police car, it would be a different Lego police car. It would have a different shape. So this is changes its shape, and that could affect its function because the most important thing in biology is shape. So here is the promoter region. The operator is within the promoter. Here's the -10. This is the +1. So this is -10. This is -35. So in this case, the operator is right in the middle, and it directly prevents the RNA polymerase from binding. And we've already covered this in a previous chapter. Remember, +1 is the start site for transcription. So anyway, this is where the operator looks like. Here's the RNA polymerase. It's going to bind it's going to recognize the 10 and the 35 and bind to it. And remember, its shape dictates where it's going to start reading. So this is the repressor, right? In the inactive form, it just kind of floats around. Uh, so this is different. When LAC-I was made, LAC-I is already in the correct form to bind to the operator for the LAC operon. Remember that? So here, trip R is produced. This is trip R is produced, but it's not active. In that shape, whenever it comes out of the mRNA and it's turned into a protein, it doesn't have the correct shape. So it cannot bind to the operator. Now, if tryptophan is present, it binds to the inactive repressor and causes it to change its shape. Now it recognizes that operator sequence, binds to it, and blocks the polymerase from binding to it. So in this case, tryptophan's present, genes are off. Tryptophan's absent, genes are on. Okay, so this is the other way that it's regulated. It's regulated by attenuation. So in attenuation, we have, um, so this is the RNA sequence. We have AUG and so on. We know that this is RNA because there are U's found in it. And we know RNA is single stranded. So unlike DNA, I mean, DNA can fold and stuff, but it's not going to, unless it's single stranded, it's not going to bind to itself because this is the backbone. There's no hydrogen bonding going on there. But RNA is single stranded, and so its bases are exposed and they can interact with each other and form these crazy structures. And we talked about this. We talked about the tRNA has that cloverleaf shape because of the interactions of the bases but with each other on that single strand. I mean, just think of it as a thread. It's not a stiff piece of wire. It's like a piece of string, and you can move it around. And you know, A's have affinities for U's, and G's have affinities for C's, and we can get these secondary structures. So I'm not going to get into row. There are different ways to terminate stuff. There's row independent, which means it doesn't need row, and there's row dependent. In this case, this is a row dependent independent structure. This is the stem loop. You can see G pairs with C, C pairs with G, and so on. And it's going to make this shape. All right. And so the the so in bacteria, this is we talked about this before. So this is co-translational. That means transcription and translation occur at the same time. So here's our gene, um, and the polymerase is on here, and it's going this way. As it's going, it's going to create the messenger RNA at the same time. And this is hard to show you, but that's what's going on. And so the ribosomes can bind on here, and they start reading. So that's what's going on here. We're going to have a ribosome. We'll have the large subunit and the small subunit, and there'll be a Shine-Delgarno sequence. Uh, in this region here, and it's going to start reading. So, it's going to, the AUG, you guys know, makes methionine, and you could use the genetic code and verify all these amino acids are correct. And so it's translating while this mRNA is being made. So it's going along here at a clip, and it's reading it and turning it into a protein. And this is the trip operon. So in this region, this is the attenuation region. In the attenuation region, there are two tryptophan codons, which are right here. And the tryptophan, so if there's tryptophan present, then the tRNA can bring in the anticodon to this codon, and the ribosome is going to just continue on its merry way and keep reading, right? And so this is relatively fast. And the ribosome has an effect on the way that this structure is made right here. And remember, this is transcriptional termination. So it's going to interfere with the RNA polymerase, not the ribosome. So in this case, translation has an effect on transcription. Translation speed can terminate transcription. And I'm not going to get into the details of this, but what you should know is that if there's tryptophan, the polymerase is just going to continue, right? So with trip, we don't need trip. There's already trip there. The RNA, not the polymerase. The ribosome is going to continue on reading the codons and making the protein. And all of these are needed for biosynthesis of tryptophan. Now, if there's no tryptophan, the ribosome is going to pause. When it gets to tryptophan, there's no tryptophan. It can't continue. So the ribosome stops. And that stoppage changes the shape, and that causes transcription to terminate. So this is just a double check. Now think about it. If there's tryptophan present, we want to make the transcription of this gene, so this is a leaky accident. If tryptophan is present, we don't want to make this gene. We want to terminate transcription. So if the ribosome speeds through there, if there's tryptophan present, it's going to kind of knock off. You can think of this as a race, right? Here's the RNA polymerase making RNA, right? It's making the RNA. Here's the ribosome, and it's reading it and turning it into a protein. So this thing is the ribosome is chasing the polymerase. Think about it this way. I'm just going to try to make it simple. So the ribosome, and it's definitely more complicated than this. So if you want to know more, take a molecular genetics class, and I definitely talk about row independent and dependent termination. But I'm not going to do it here. Let's just think about this ribosome. If this ribosome catches this polymerase, right, just like if someone's chasing someone with the football, they're going to tackle them and they're going to knock them to the ground. So that polymerase gets knocked off and no more transcription can occur. So if there's tryptophan present, the ribosome just smoothly goes through those double tryptophan codons and changes the shape of the stem loop and the row independent terminator and ends transcription. Now, if there's no tryptophan present, the ribosome pauses. Just think about the runner stopping whoever has the football and stops chasing them. Well, it's going to get to continue on to the end zone. And so the polymerase is going to continue on producing each of these RNAs, and then those are all going to get read into proteins. And in this case, That makes sense because if we don't have tryptophan, we need to make tryptophan. That's how attenuation works. All right. So these are the stem loop structures that are produced. If tryptophan is low, the ribosome pauses, right? Then this domain 2 and 3, they bind to one another. In this shape, right, in low levels of tryptophan, this is not going to terminate the polymerase, right? So the ribosome, remember, has an effect on the RNA polymerase, and so it will not terminate the RNA polymerase until we get transcription occurs. Now, if there are high levels of tryptophan, it can speed through this, right? It's not pausing. So it ends up interfering, it's blocking domain 2. If it's on domain 2 cannot bind to domain 3. So what happens is these domains down further make hydrogen bond with each other, and they make a different shape. So if domain 3 and 4 um, create this stem loop, then this does terminate the RNA polymerase and it stops transcription. Okay, so that's how it works. I'm not going to get into the details of it, but basically, what I would want you to know for the test is at low tryptophan levels, domain 2 and 3 bind together. There's 4 domains. 2 and 3 bind together, and in that shape, transcription does not terminate. In low levels of tryptophan, we want the transcript, we want the protein made, we want more tryptophan. High levels of tryptophan, it interferes with 2 and 3 binding with each other. 3 and 4 now bind with each other because that ribosome is just kind of screaming through that RNA. And so in that shape, where 3 and 4 bind to each other on the RNA, then that causes the polymerase to stop transcription. And so that terminates, and this is just a double safety for that, right? We still have the trip R, binding to the operator and can repress that system. Okay, so this is just another slide of exactly what I just said. This is just showing you a different version of it. I'm not going to go over all this. This is what it actually looks like. So remember we talked about the cloverleaf structure that is drawn, and that's this is just detailed what's going on here. This drawing here is my drawing of a ribosome, so I know it's terrible. Okay, so in arabinose metabolism, arabinose is another sugar that can be used as an energy source by bacteria. Um, we don't have time to go through the arabinose operon, so I'm not going to cover it. But I just wanted to show you. I'm not going to cover it in detail. I just want to show you that. Um, so even though, and this is a piece of DNA. So even though there are things that are far away from one another, it's and this is because DNA is a double helix. We draw the double helix like so, but it's not like a perfect helix. It's more like this. And so you have like a major groove, which has got a lot of space, and a minor groove, which doesn't have a lot of space. So in the major groove, proteins can interact with the bases and look and see what they are. So even though we have DNA, which is double stranded here, and we have proteins, right? Remember, these are trans factors transacting factors can bind to DNA that's far away by bending it. So imagine you have a string and you're bending it, and then you have two regions that recognize the DNA. So in this case, this recognizes a sequence of DNA. This recognizes a sequence of DNA, and these can actually bend the DNA in a stem loop. So you can have things that are really far away. We're going to talk about this in eukaryotic gene expression uh, in detail. But I just want you to be aware that DNA is flexible, it can move, and you can have things that are very far away from the promoter, that tens of thousands of bases that can affect transcription of that gene. And that's all I'm going to say about arabinose. It's just a different way of regulating it. I think if you know lac and you know tryptophan, and I'm certainly not going to have time to go into this. So we're going to skip these two. Um, it won't be on the test. I put this in here because I thought it would be kind of interesting. Uh, because it does a lot of weird antisense left and right-handed thing, and this kind of demonstrates that. I know you guys, you're not the only ones. A lot of people, look at DNA and they're like, okay, DNA is double stranded, and this is an A and this is a T, and so on and so forth. And so, the polymerase is only going to read this direction. But remember, you have another strand of DNA, and it can be read in the opposite direction. As long as there's a promoter there, the promoter elements, and it can be read uh, from 3 to 5, so it's written 5 to 3 always, but it can be read 3 to 5 on either one of those strands in either directions. And this is a good example of this. So this gene is N gene is being produced by reading in this case, it would be the upper strand, right, in this direction. And this one, so this is an operator right, and it's making this crow product, and it can go in the opposite direction. So this is the lower strand. Um, and this is done a lot in viruses because they only have, 40 to 50,000 nucleotides. So all of their genes are super highly compressed, and they utilize both the upper and lower strands uh, for producing their products. So that's really all I'm going to say about that. And if you're interested in this, you can look it up. It's kind of a fascinating system. Um, I am going to say a little bit about uh, the lytic process. So let's just talk. We're not going to talk about bacteriophage lambda, but we talk about lysis and lysogeny. So when scientists study bacteria or viruses, these bacteriophages, viruses are very specific for what they infect. So you're probably not going to get a virus that's a dog virus in a human because they're real specific for what they infect. So if they infect a bacteria, chances are they're not going to be able to infect a human or a dog. So when we're talking about these phages, we talk about phage. When you say the word phage, that usually is a virus that infects a bacteria. So sometimes we call it bacteriophage. Some people use the word phage as just virus. But when I hear the word phage, I almost always automatically think that's a virus that infects a bacteria. Now, to look at phages, uh, we we smear bacteria that they like to attack, I guess, onto a petri dish. So there's agar on here, and there's a lawn of bacteria. Let's say it's E. coli, just it's easy. And so if we infect this with a virus, the virus is going to find, that's a terrible color for this. Let's use green. So so let's say the virus infects, it's still a terrible color. Like red is just a lot. Okay, so you're going to get these areas of clearing because what happens is you have your bacteria, it has a cell wall, and the virus, bacteriophages look like kind of like lunar landers, and they're going to come in and inject their genetic material. They hijack the hosts, and the hosts make lots of viruses. They fill up with viruses. Right? They have all these viruses in them, and then they explode, and that goes on and infects the next one. So you have one cell, and it radiates outward. And so generally you get an area of clearing, an area where there's no bacteria. Oh, just erase the whole thing. So there's an area with no bacteria at all on it. So there's this still a lawn, it's cloudy. And you get this clear area. And that's called a plaque. And that's the area where the virus has killed the bacteria and made, more viruses. So when a virus inserts its DNA into a bacteria, it can do two things. It can do something called lysis, which is this, where it will infect, make its progeny, break open the host, and then go on to the next. So if you see a plaque, you know that this is a lytic phase, not phage. This is a lytic phase. So it's the virus is undergoing lysis, right? It's actively infecting neighboring cells and destroying the bacteria. But there's another way it can go, and this is called lysogeny. So in lysogeny, after the DNA is inserted from the bacteria, it goes into the host, integrates into the host DNA, and just sits there. So here's the chromosome. Here's the chromosome for E. coli. This is its chromosome. And the phage DNA can insert itself in here. And then every time the bacteria copies itself, we get a new bacteria. It's just hanging out in the host. But as it's doing that, it's every time the host bacteria the virus is lysogenic in it, it's going to get passed on to all of the daughter cells that are produced by binary fission. Okay, so there's no progeny made, there's no lysis of the host, it's just hanging out, and it's making more viruses as the bacteria is replicating. So those are the two ways. So there's a lambda phage that usually infects E. coli. This is the virus, this is the host, right? This is the bacteria, this is the virus phage. Okay. So that bacteriophage can do either. It can be lysis or lysogeny. So this is just showing you what I just was trying to draw. So this is the virus, the little spaceship. It lands, it infects, it makes more, and then it breaks open. Here, this is so this is lysis. This is lytic growth. This is lysogenic growth. So it just enters into the bacterial chromosome. It's sad for me, actually. Anyway, and then these are the plates, right? So this is also a kind of a bad drawing, but the lysogenic cells are not going to have a clear area. So, I mean, it's really hard to like show this, but it wouldn't affect, you would still have a cloudy lawn of bacteria. If it's lytic, like we talked about, you'll have these clear areas that have no bacteria on them. And that's how you can kind of tell if it's lytic or lysogenic. All right. So how does the phage decide to do that? It's kind of a good strategy. So if in humans and animals, the virus integrates into the host uh, when times are good. Um, because it doesn't want to kill the host, right? But if like there's a stressor, like maybe the host is sick or whatever, something's going on, maybe it's not getting the proper nutrients or whatever, the virus uh, basically says, "Oh well, I better jump host because this one's gonna die." But in bacteria, it's the opposite. So in bacteria, viruses want to be lytic when lots of nutrients are around. And this is because bacteria their cells can separate, right? They go their own way. In animal host, we're multicellular. So, our lung cells cannot just like take a trip down the river like bacteria can. And so when times are good, they're going to make as many viruses as they can because they know that there's a lot of cells around because bacteria are only going to divide when there's lots of nutrients. So if there's lots of cells next to them in bacteria, it means that they're good times because the bacteria is dividing. And so in good times, they're going to want to go lytic. They want to infect as many bacteria as they can because in bad times, there may be no other bacteria around. And so if they went lytic and they burst out of the cell, they may not find another host uh, that they can jump into immediately. So bacteria are different than humans and animals as far as lytic and lysogenic. So in bacteria, good times lytic, bad times lysogenic. So this is the genome. I'm not going to test you on any of this stuff. Um, but remember, this thing is only like 50,000 nucleotides. So it has a ton of genes, and these genes are all stuffed together, and they may be on expressed to the left or to the right or eat both because it doesn't have a lot of DNA, a lot of space to do this. Um, all right, so this is going to get kind of into the specifics of this, and I'm not gonna ask you this on the test. We don't really have time to go over this because it's let's see what we have to do. Okay, so I'm just kind of looking at the schedule, and we still need to cover eukaryotic gene regulation, and we need to cover um, developmental genetics. We're not going to get to transposons. We only have 2 more weeks. So next week I'll do lectures on eukaryotic gene regulation, and then the following week I'll do lectures on developmental genetics, and then that'll be the end of it. And then we'll have your final exam the following week. So that's the plan. I'll do lecture 19 next week, and then lecture 20 the following week. So I'll probably split them into 2 lectures. Um, the first part of this prokaryotic gene regulation was a little long, so I'm going to cut this one short. It's only 40 minutes, and that's probably perfect. Again, Nobody's come to office hours, so if you need help, I'm here. I'm available to help you during office hours. You have all kinds of other resources to help you. Uh, the MCC tutoring. You have tutoring from Canvas. You can send me an email. You can talk to your fellow classmates. If you have questions, right, there's a discussion board. You guys can discuss this stuff. Uh, so, anyway, if you have any questions, let me know. Send me an email. Come to office hours. Otherwise, I will see you at the next lecture.